friends, today we will try to solve the problem related to genetics, and the problem is the RFLP pattern observed for two pure parental lines denotes as P1 and P2, and their F1 progeny is represented below. And this P1 plant has red flowers and P2 has white flowers. So F1 progeny was backcrossed to P2, and the result obtained showing the number of progeny with red and white flowers, and their RFLP pattern is also represented below. So the table shows RFLP bending patterns of P1, P2 plants and their first filial generation, as well as F1 backcrossed with P2 and all the resultant F2 progeny, how many numbers they will get and what are the bending positions. Okay, and then after, from this table there are certain conclusions are made, and among that we have to decide that which conclusion is right for this problem. So the first is the DNA marker and the gene for the flower color are 10 centimorgan apart. The second is marker and gene are 5 centimorgan apart. The third one is marker and phenotype are independently assorting. And the fourth one is marker and gene for the color segregate from one another. So now from the problem we can clearly get a solution that P1 has a red flower, P2 has a white flower, and there is cross between them gives a first filial generation, and all the first filial generation has a red flower. Now there is an back crossed with F1 and P2 contains red as well as white flower. So all F2 generations means we get 4 gametes of F2 generation. And what are the bending positions and what are the number of positions of all F2 generation, that is given in the table. So from this table we again understand that RFLP bands of P1 is 1, P2 is 2, but this position of this RFLP band in P2 is different than P1. Then after, F1 progeny contains 3 RFLP bands. Now there is an backcross with F1 and P2 generation give rise to 4 F2 progeny, and from this position of band we can clearly see that in first F2 generation and fourth F2 generation the bending pattern of RFLP is same as the F1 generation, and there is slightly different in second as well as third generation of second filial, and they contain only 2 bands of RFLP. Means 2 progeny looks like parent, and we can say that these 2 progeny, so first and 4 are parental phenotypes, and second and third are recombinant one. So if we consider this is explanation of what are parental and what are recombinant types. So from this table we can see that what are the number of parental phenotypes. Number of parental phenotypes are 45 plus 5. So total 50 are looks like a parental, and rest of the remaining what are 5 plus 45 are recombinant types. So the ratio of recombinance versus parental, that is 1:1, and this 1:1 ratio clearly mentioned that the genes are independently assorting, means they are following the rules of Mendelian genetics, independent assortment as well as segregation. So here the question is that how we can identify that the following genes can follow a linkage rules or as Mendelian rules, means the genes are linked or not. So clear solution for that is that if parental as well as recombinant ratio is 50%, then the genes are assorting independently. But if recombinance and parental ratio, parent phenotype should be more than 50% in case of linkage. So if number of parent are greater than 50, then it will follow the linkage rule, means the genes are linked with each other and they are not following the Mendelian rules of genetics. But if it is 50% or else less than 50%, then there are chances that the genes are not recombinant one. And the second assumption is that recombinants are always 10% or less than 10% in case of a successful linkage between the remaining genes. Okay, and sometimes it can be greater than 10%, but the rules for the parental genotype should be greater than 50%. So if parents are 60% or rest of the 40% are recombinant one, then we can say that yes, it may follow linkage rules, means the genes are linked with each other. But if the parents are 50 and recombinants are 50, then it will again follow 1:1 ratio, that is Mendelian rule of genetics. So obviously the distance between the marker gene and the flower gene is they are distantly aparted, okay, is higher. So from the assumptions we can clearly say that two distance are given, that are 10 centimorgan and 5 centimorgan. So obviously distance can't be in less between the marker gene as well as our target gene in case of Mendelian genetics, because they are following Mendelian rule of genetics. So obviously distance should be 10 centimorgan. And our assumption number 1 is fulfilling this condition. So what assumption 1 clearly states that the DNA marker and the gene for the flower color are 10 centimorgan apart. So option 1 is the correct answer for this problem. So I hope you will solve such kind of problems in a easy way. So please like, subscribe and share to my channel. Help me and continue